that can see that I'm in a way cursed definitely for the last decade with always multiple affiliations, which makes it more difficult when you register for conferences or something, when the text box where you put in your affiliation is somehow word limited, right? Okay, today I would like to talk a little bit about our work on comparative approaches to link phenotypic differences to differences in the genome. And I would like to start with what I personally find most fascinating about biology. And this is essentially nature's spectacular phenotypic diversity. And I try to illustrate this here with species that can echolocate and can navigate in complete darkness, sperm whales that can dive for more than 1 kilometer deep in the ocean and can dive for more than 1 hour. We have birds that can hover on the same spot. We have species that have lost eyes and limbs, and other mammals that have evolved body armor convergently. And I think this diversity is potentially what Charles Darwin meant when he concluded the origin of species by stating that endless forms, most beautiful and most wonderful, have been and are being evolved. And in the genomic era, all the species that are shown here have sequenced genomes. And of course, by now we have in the order of maybe 500 mammalian species, and definitely about probably 400 birds have fully sequenced genomes. And because many of the fascinating traits here have a strong genetic basis, we can now start addressing the question, what are actually the genomic underpinnings of these phenotypic differences that we see among these species? And that has pretty much been the central question that my lab has been pursuing in the last decade. And so if you think about this question, what I like to do is I like to distinguish conceptually different types of genomic changes that can affect the functional repertoire of the genome and can therefore contribute to phenotypic changes. For example, changes in regulatory elements can lead to differences in expression patterns of genes. You can have changes in the protein coding sequence, which can alter protein function or result in a novel protein function of a conserved gene. Novel genes can arise in evolution, for example, by gene duplication, but also de novo from previously non-coding sequence. Sorry. Okay, can you see back the PowerPoint?
Yes, yeah. All good.
Good, okay. Sorry, when some updates in the background happen. And of course, ancestral genes can also get lost in the course of evolution. Of course, there's this is not a complete list of the conceptual types of genomic changes. But those are the 4 kinds of changes we worked on extensively in the past. And in this talk, I decided to, in a way, talk about 3 of those 4 types of changes. So in the first part, I will talk about differences in regulatory elements and eye degeneration in subterranean mammals as a phenotype. In the second part, I will tell you about the importance of gene losses and that they could contribute to adaptive evolution. And in the third part, I will talk about convergent changes in the proteins. Okay, so for the first part, I would like to start by explaining how we find association between phenotypic and genomic changes. And to this end, now I can almost say back in the days, we have developed a so-called Forward Genomics framework. And the idea is that we can start or look at ancestral traits that have been changed or lost in several independent lineages. And then we can conduct a genome-wide screen. So here, the horizontal line represents in a way the stretched out genome. And all these boxes represent regions which are overall well conserved between those species. And we can screen all these conserved regions to find those which are more diverged in the sequence or maybe even completely deleted in those species where this ancestral trait has been altered or has been lost. And I will give you a few more details of how we find and search for these associations. So let's focus now on one of these conserved regions. So typically this is a few hundred base pairs in the genome. This is the phylogenetic tree of these species. And given the sequence of this element in the extant species, we can computationally reconstruct all the ancestral states, which are the internal nodes here in this tree. And with these reconstructed ancestors, we can now focus at the beginning and end of every branch, and we can measure how much of the sequence was conserved along this branch here as an example. And in this toy example, we see that 11 out of 15 bases here are identical. So we would say 73% of that sequence has been conserved along this particular branch. And then we can color or classify all the branches into two groups, into those along which this trait was likely lost, here shown in red, and those in green where the ancestral trait was largely preserved. And so with this, we now have two groups of branches. And for each of them, I can measure a per branch conservation value. And we can now ask the question of whether the trait loss branches are associated with lower conservation values or more sequence divergence compared to the trait preserving branches. And what I would like to point out is that this species and this phylogeny, of course, are all related. And that is simply what you can see in the tree. But if I dissect this phylogenetic tree into the branches, then each branch, and therefore each of these measurements here, represent independent evolution. So this is essentially independent data here. And therefore I can do something quite simple. I can, for example, use Pearson correlation and ask whether I find this observed, this trend here that the trait loss branches are associated with higher divergence values. And from this, I can compute a p-value, whether there is a significant correlation. And if so, we would say that we found an association between this phenotypic pattern and the sequence divergence in this particular genomic region. And then essentially the method is to walk over all these conserved regions we have, or we can detect in these multiple genome alignments. So let's put this into practice. One trait that I was always very intrigued with is the loss and reduction of vision in subterranean mammals. And if you look here at this simplified mammalian phylogeny, you can see that 4 independent lineages have adapted to this underground environment. And one characteristic feature of this is a high degree of eye reduction or eye loss. And you can see that those two species here, they actually dig tunnels with their forehead. And this likely is the reason why the rudimentary eyes, which they possess, are buried behind skin and fur. So they are truly blind. So we were interested in what are the genomic changes that are associated with eye degeneration in these subterranean mammals. And as I said, Forward Genomics as input can consider conserved genomic regions, and they largely fall into 2 classes. In mammalian genome alignments, the smaller part are actually exons of conserved coding genes. And so if we apply Forward Genomics and ask for increased sequence divergence in those subterranean mammals, then we find, I think it was in the order of 208 or exons belonging to 208 coding genes. And to make a long story short, those genes are highly enriched in having eye-related functions. And the work by our and many other labs have shown that these subterranean species have lost many genes which have functions in photoreceptors, in the lens, and in eyes in general. The larger part of these conserved genomic regions are the non-exonic elements. And they are interesting because they often overlap cis-regulatory elements that determine when and where and how much genes are expressed. So in these genome alignments that we constructed, we got in the order of 350,000 of these conserved non-exonic elements, and running forward genomics on them, we found significant sequence divergence in the subterranean mammals for about 9,300 of them, so 2.7% of the total data set. And so this, of course, raises the question of what is special about this diverged set of non-exonic elements. And that's a question which is not super straightforward to answer, because in contrast to genes, where we typically have functional annotations, we often have mouse knockouts, we have Go terms. For those non-coding elements, we typically know much less. So we have been pursuing two statistical approaches to investigate what is special about this diverged set here. And both approaches assume that many of these elements have cis-regulatory function. And so in the first approach, we associate each of these conserved non-exonic elements with potential target genes that could be regulated by them. And for the very most part, this is often the gene upstream and the gene downstream. And then with these associated genes, we can ask if they are enriched in certain functions. And to make a long story short, I will not show you the raw data here, but in a way just summarize it. We found that the conserved elements, which are significantly diverged in those subterranean mammals, are highly associated with eye and in particular lens-related genes. So they tend to be in the proximity of lens-related genes. A second, probably more direct way of assessing what is special about these elements is to use functional genomics and, for example, obtain a set of eye regulatory elements from, for example, model organisms. And we can then determine how many of these diverged non-exonic elements overlap these eye regulatory data. And we can compare this overlap to the remaining non-diverged elements. And with this, we can, in a way, construct we can do a Fisher's exact test to quantify whether the diverged elements have a tendency to overlap these eye regulatory data sets like eye enhancers. To cut a long story short again, we've been using both publicly available data sets from the adult retina or adult lens of mice. And we've also been using a very powerful assay called a TAC-seq to create new data sets for the developing eye of mice, which were lacking at the time. And as shown here in these bar charts, after correcting for multiple testing over different sets of regulatory elements, we found that these diverged non-exonic elements have a significant tendency to overlap these eye regulatory data sets. So these, I said many of these non-exonic elements overlap regulatory elements. And we know that regulatory elements consist, of course, of DNA bases. But what's more important are binding sites for transcription factors. So proteins that bind to specific motifs in these sequences and then confer the regulatory properties of this element. And we can predict these binding sites using in a way, motif. So those logos represent what certain transcription factors tend to bind. And this gave us the idea that when we are investigating regulatory elements, then we can probably enhance both the sensitivity as well as the specificity in this genome-wide search when we consider binding site divergence and not so much sequence divergence. And I would like to illustrate what we were thinking at the time with, in a way, two simple toy examples. So here we're comparing the DNA sequence between, say, an ancestor and a species, and you see that only at 2 positions a substitution happened. So at the sequence level, there is barely any divergence visible. But if we look at transcription factor binding sites, we would see that both of these changes would destroy potentially important binding sites. So this is something which would not be visible in the standard forward genomics screen for sequence divergence, but it would be visible if we screen for binding site divergence. And this would be a case where we would detect or would increase our sensitivity. In this example here, we have much more changes at the sequence level. But if we look at the binding site level, we realize that some of these changes are actually outside of the binding sites. Or in this case, I try to illustrate a substitution that is still compatible with binding, because here this transcription factor tolerates both an A and a T at this position. And in this case, we have a case of binding site turnover. So this binding site here is destroyed by this mutation, but over here, an equivalent binding site is created by additional mutations. So if you look at the binding site level, then not much happened. This is largely conserved, and therefore the functional properties of this putative regulatory element are likely also conserved. So not catching such a case in a genome-wide screen would likely increase specificity. And so what we did was we implemented a method that we call regulatory element forward genomics, or REforge. And this is based on the exact same framework of reconstructing ancestral states and having two classes of the different branches. And now, instead of measuring for every branch the sequence conservation, we are computing a score that measures how well are transcription factor binding sites conserved. And so we applied this method to the exact same set of non-exonic elements, and all what we're using as additional information were the binding motifs of about 20 transcription factors that, based on literature, have well-known functions in eye development and function. And so this gave us significant binding site divergence in these subterranean mammals for 3,700 of these elements. And so compared to the amount of non-exonic elements that we are detecting increased sequence divergence, this is of course much fewer, indicating that we have a higher specificity. And furthermore, despite having a smaller data set, we actually found stronger enrichments with the overlap of these functional genomic data sets that measure or determine eye regulatory elements, indicating that in a way we have an increased sensitivity. And indeed, we found binding site divergence in several interesting regulatory elements near key eye genes, also genes that control eye size, which could be relevant for the phenotype. You can actually see here that the rudimentary eyes which are present are really tiny. So to summarize this part, we found that there is both a striking signature of sequence as well as transcription factor binding site divergence in a number of regulatory elements. And this suggests that not only losses of eye-related genes, but also divergence in many eye regulatory elements contributed to these eye degeneration phenotypes, which characterize these subterranean mammals. And for one of these elements, we actually did further experiments. We were focusing on a lens regulatory element, so something that showed up as a lens ataxic peak in our data sets. And this element had a number of mutations in the blind mole rat. And what we did was we were introducing the exact mutations into the mouse. So essentially replacing the orthologous mouse sequence with the sequence of the blind mole rat with CRISPR. And in this mouse model, we found that the target gene, which is a key lens gene, is substantially upregulated during embryogenesis. And this indicates that, together with luciferase assays we did, that this is actually a repressor element, and that the mutations which happened in the blind mole rat destroy the repressor function. and therefore leading to upregulation of the gene. Only, this is something I have to say, only during embryogenesis. Okay. In the second part, I will now switch to talking about losses of genes. And the motivation for this project is similar to the motivation for the REforge method, in that the standard forward genomics approach that I explained is on the one hand very nice, because by looking at sequence divergence, we can run a comprehensive genomic screen that where it doesn't matter what these conserved elements actually do. They can be parts of exons, which I showed you, can be parts of regulatory elements, can be something else. It doesn't matter, as long as it's conserved, we can quantify sequence divergence. The downside is that we are ignoring, by treating all these elements exactly the same, we are ignoring important information we have about the different classes of elements. And I just showed you that if we know this is a regulatory element, then we can do much better by looking at binding site divergence. And of course, if you think about coding exons, we also know precisely which mutations actually lead to divergence and even loss. So here I'm showing you a mini exon, so to say, with a splice site and a reading frame. And we know that not only, of course, entire exon deletions, but also many more subtle mutations, like a single base deletion shifts the reading frame and leads to completely different, typically truncated protein downstream of this site. We know that mutating these conserved splice site dinucleotides in mammals and vertebrates in general lead to exon skipping. So you would lose that exon at the level of the transcript processing. And we know that certain substitutions like this one can create in-frame early stop codons and therefore also highly truncated proteins. So those are all mutations which are clear evidence for gene loss. And so our goal was to identify these gene-inactivating mutations across many species and at a very high accuracy. And if you look at this picture here, this may appear as a rather trivial problem. And this is actually precisely what we thought when we started this project. But if you want to do this job across, now we are on the order of hundreds of species where we're applying such methods to, and at a very high accuracy, because you don't want to manually curate, or you want to manually filter out lots of false positive, then it turns out this is actually quite a challenging problem. And it took us several years to build a pipeline that addresses a number of issues related to the quality of the assemblies, that they are incomplete, the underlying issues with the underlying genome alignments, which we have been using as a basis, but also the fact that the exon intron structures of conserved genes can evolve. And this makes it difficult to, in a way, detect whether the gene is still conserved or actually whether there's evidence of gene loss. And I would like to illustrate maybe the two predominant issues that we were running into. And this is now not toy data anymore. This is a real example aligning the human genome here to the cow. And you see that this acceptor splice site is clearly inactivated. AC cannot work as a U2 intron acceptor site. So this would be evidence for that this exon is likely skipped and that the gene is not intact. While the gene is annotated in the cow, and it turns out the splice site that's annotated is over here. So it has shifted exactly 9 bases upstream. And as a result, all what happened is that this exon now has 3 additional amino acids. Another case is this here, where I would like you to appreciate that if you look at the identity of this sequence alignment, this looks really good, right? And we see a 4-base insertion in the cow, so something that would shift the reading frame. Again, this gene is annotated, and we were wondering, why is this actually? And it turns out you can find an alternative alignment, which should not be confused with alternative facts. This thing really, in a way, you can construct it, right? It really exists. And here, this 4-base insertion can be shifted to an intron position, because essentially what happens here is that we have a microduplication. If you would get to see such an alignment, you would say, well, there's no problem with this exon. It's fully conserved, splice sites are intact. With this alignment, and this is what the genome alignment actually looked like, we would conclude there is a frame-shifting insertion, evidence for gene loss. So to make a long story short, we were building methods and filter steps to cope with all these problems as best as possible, and integrated this all in a gene loss pipeline. And we were testing the specificity. So in a way, taking conserved genes, a large set of conserved genes, I think it was in the order of 12,000 genes and more, and asking how many of them are falsely classified as gene loss. And we obtained a very high specificity of more than 99%. And so with this pipeline, we felt that it's good enough to be systematically applied. At the time, we've been applying it to a whole genome alignment that we have constructed containing 62 different placental mammals. Right now, we're in the process of, just to give you a feeling of how fast genomes are accumulating, we're doing in a way a similar job now over 450 different placental mammals. So we in a way systematically created gene loss catalogs for all those species shown here. And that was a great data set to investigate lots of different scientific questions. For me, what was most interesting to do was to look at genes that are lost in those species that have obvious prominent phenotypic adaptations. And that's something I would like to talk about in the following. And so I would like to take a step back, and if we think about which of these different genomic changes is likely contributing to adaptation, then I guess we would intuitively say that it's those novel expression patterns, novel protein functions, novel genes that are most relevant. But we found evidence that also losing genes can contribute to adaptive phenotypes. And this is a little counterintuitive, because essentially it means that you take a hammer and you break something that already exists and is working, and somehow this should be good, right? But actually in the bacterial world, based on artificial selection experiments, this principle is well established. And I would say among mammals, there are a few pretty well understood cases related to immune receptors, that losing those receptors prevent certain pathogens to enter the cells. But apart from that, there were, I would say, few cases of potentially adaptive gene losses known. And so what I would like to do is I would like to distinguish two major reasons that can explain why a gene got lost in the course of evolution. And those two principles are use it or lose it, and less is more. And I would like to explain both of these using those two genes here. And those two genes are exclusively lost in the sperm whale and intact, as far as we can tell, in the genomes of all these other mammals. So the first gene is BCO1, and this encodes an enzyme that is able to cleave beta-carotene into vitamin A. So losing this enzyme suggests that the sperm whale is unable to synthesize its own vitamin A. And that was actually quite unexpected, because based on old literature, the internal organs like the liver are incredibly rich in vitamin A. So where's this vitamin A actually coming from? We believe it's likely directly coming from their diet, because what they do, or what they hunt at several hundred meters down in the ocean, are actually these medium-sized or large squid. And these squid contain very little beta-carotene, so little substrate, but they provide already plenty of vitamin A. So we believe BCO1 was lost as a consequence of this dietary adaptation, because the sperm whale essentially, after adopting this specialized diet, had little or no use to maintain this gene here. The second gene is called AMPD3. This caught our attention for being highly expressed in erythrocytes, so red blood cells. And we found that's interesting because in contrast to most other cetacean species, the sperm whale is an exceptional diver that can dive for more than 1 hour to depths of several thousand meters. Based on mouse knockout experiments, we actually know what happens if you inactivate this AMPD3 gene. So if you knock this out, then the ATP level in those erythrocytes, they increase. And this puzzled us for a long time because ATP is typically well known as molecular energy, right? But what I for a long time didn't know is that ATP is also an allosteric effector of hemoglobin, and it specifically stabilizes the hemoglobin form that is not bound to oxygen molecules. So if ATP levels increase and you stabilize the oxygen unloaded hemoglobin, then essentially you're shifting this curve that relates the hemoglobin oxygen saturation to the partial oxygen pressure. And this essentially means that you facilitate oxygen release in the capillaries. So in other words, by losing this gene, you're facilitating oxygen transport from the lung into the tissue. And we believe that can certainly be an advantage for such a long diving species. So the loss of AMPD3 could be an example where for the sperm whale, less genes in this case is more with respect to this particular phenotype. And so by observing this and a number of other lineage-specific gene losses that could be adaptive in certain lineages, we thought that if evolution comes across this solution once, then it may come across the same solution in other lineages that maybe have similar adaptations. And the reason for thinking that is that random mutations, so with random mutation, and that is the substrate of evolution, it is typically easy to break something, while it's much harder to create something new. And so what we did was to, in a way, investigate whether there are convergent gene losses that could contribute to convergent adaptations. And this is actually exactly what we have started this project for. So now we're able to run this Forward Genomics pipeline, not focusing on sequence divergence, but we can now use those gene loss catalogs directly and associate these convergent traits with convergent gene losses. The specific phenotype I would like to talk about is dietary specialization in mammals. And so here, what we did was we were focusing on strict herbivores, and we're asking which of those systematically, which genes are preferentially lost in independent herbivore lineages. And we did the same with carnivores, but the most striking example we found relates to a repeated loss of a gene, PNLIP-RP1, in independent herbivores. And in fact, this gene is lost in 8 independent herbivore lineages. The gene is related to the digestion of dietary fat, triglycerides, and this is actually done by the triglyceride lipase, so pancreatic enzyme. And this enzyme is only active if it's bound by a cofactor. PNLIP-RP1 has no enzymatic activity, but it has the same binding pocket for the cofactor. So in other words, this is dead weight that can bind and sequester and steal the cofactor and therefore competitively inhibit the actual lipase that digests dietary fat. And fortunately for us, this effect was already in a way well studied in knockout mice, where you can see here in this chart that on a regular diet, the fat mass of these knockout mice essentially doubles. So losing P and LipRP1 is a way to improving the efficiency of digesting dietary triglycerides. And we believe, given that the plant-based diet which these species here consume is rather limited in fatty acids or fat, that this is certainly an advantage. And therefore, this could be a case of convergent less is more. So to summarize the second part, we developed quite an accurate method to detect gene losses in genome alignments. And we believe that the vast majority of the genes that we detect can be explained by this use it or lose it principle. So meaning not having selection to maintain that gene anymore because its function became obsolete. Nevertheless, exemplified by the sperm whale and vitamin A, such gene losses can provide novel insights into species biology that would be very hard to get otherwise. And we think sometimes it can also be an advantage to lose certain genes, and then the loss of ancestral genes can also contribute to adaptive evolution. And in addition to those two examples I presented, we found other genes that are involved in epidermal, have functions in the epidermis, of physiology, which could contribute to epidermal and physiological adaptations and cetaceans, partially convergently lost with manatees. We found a number of genes that work as, in a way, built-in brakes, and they limit insulin sensitivity and insulin secretion, and observing that these genes are specifically lost in fruit bats that have a high insulin secretion capacity and high insulin sensitivity. We believe those gene losses could also contribute to these phenotypes in these special bats. Okay. In the third part, I will now talk about changes in the protein sequences and the an aspect of convergent evolution of echolocation. So one trait, one fascinating convergently evolved trait is the ability of bats and dolphins to echolocate, which they use to navigate and hunt in complete darkness, essentially, or very turbid, cloudy waters. Echolocation requires very exquisite hearing capabilities because these echolocators emit high-frequency sounds, which are best suited to detect small objects, and they need to precisely listen to the resulting echoes. To gain insights into the genomic underpinnings of these high-frequency hearing adaptations, several groups investigated candidate hearing genes, and in a gene called prestin, they found a striking pattern where at position 7 in the protein, the exact same amino acid change from an ancestral asparagine to a threonine happened in two groups of echolocating bats and in echolocating toothed whales. And later experiments were able to show that if you change this single amino acid, then this causes a functional shift in the protein that in a direction that is consistent with adaptations to high frequency hearing. So motivated by this spectacular example of molecular convergence at the amino acid level, we implemented a method to systematically screen for such parallel amino acid changes illustrated here, and focusing on bats, echolocating bats and dolphins, we found 409 such proteins that have at least one of these convergent amino acid changes. And to our surprise, we found that those genes here, they have statistical enrichments in muscle-related terms. Specifically, we found functional annotations related to motor activity. So that there are four genes which are driving these muscle-related functions. Two of them are components of myosin motors here. Then we have an ATPase, which transports calcium in the muscle cells into the sarcoplasmic reticulum, and a gene called calsequestrine, which is a major calcium storage protein. And in total, there are 7 of these convergent amino acid changes in these 4 proteins. And what was interesting is that these genes are all highly expressed in fast muscle fibers. So how could this be relevant for echolocation? And for this, I would like to show you a slow motion video showing a bat catching a prey. And I would like you to pay attention to the echolocation clicks. So what you could hear is right before the bat actually catches the prey, it drastically increases the rate of these echolocation calls up to 200 times per second. And interestingly, this is precisely what dolphins also do. And people believe the reason those echolocators show this behavior is that they can then adjust their interception path to escape maneuvers of their prey. Because if you emit very rapid calls and listen to the echoes, which will also appear very soon because you're close to the object, right? You have precise information about the location of the object, and this will increase the capture rate. These echolocation calls, each call is produced by the contraction and relaxation of a sound-producing muscle. In bats, we know this is located in the larynx. And experiments have shown that indeed this muscle is able to contract up to 200 times per second. And that is an order of magnitude faster than the fastest locomotor muscles can do in mammals, including us. And this is why these special sound-producing muscles are called superfast muscles. Thanks to Manfred Kössel, we got actually tissue samples of this cricothyroid muscle from an echolocating bat, and with RNA-seq, we could show that all the 4 proteins or genes I was showing you are expressed in this muscle here. And so what is actually required to make a muscle super fast? And here, looking at a beautiful review summarizing work that Lawrence Rome and other groups have done in the past, one key aspect is that these muscles possess fibers with a very high shortening velocity. And this is achieved by having myosin motors with a very fast cross-bridge detachment rate. So in a way, letting this actin filament go, because that is the rate limiting step in the cross-bridge cycle. And so this is something where maybe the changes in the protein changes in those 2 genes could contribute to. The second key aspect is that those muscles have extremely rapid calcium transients to switch a muscle on and off very rapidly. And this is something where this ATPase and the calsequestrine protein could contribute to. So for this calsequestrine, we were able to do functional experiments together with Chu Li Kang from Washington University. And we could show, summarized here in this plot, that calsequestrine of an echolocating bat and a dolphin is able to form calcium sequestering polymers. And this is the major mode how this protein is able to sequester up to 60 or 70 calcium ions per protein unit. So they form in a way chains and use negative, also positively charged calcium in a way as glue. And so somehow the protein of the echolocators is able to form these calcium sequestering polymers at much lower calcium concentrations than the protein of other non-echolocating mammals. And one of them here actually was a non-echolocating bat. are able to do. So this is functional protein convergence, which is likely relevant and could contribute to superfast muscle physiology. So to summarize the third part, we found a pattern of in convergent amino acid changes, predominantly in 4 muscle-related genes, and changes in these proteins could contribute, likely among many other changes, to the physiology and evolution of these superfast sound-producing muscles which exist in these mammalian echolocators. And with that, I've reached the end, and I would like to thank all the people in my group. I've highlighted here the people who did or worked mostly on the projects that I was explaining. Unfortunately, this is not an up-to-date image of the lab because due to Corona, we haven't managed to take in a more up-to-date group photo in the last year, but hopefully when we all get vaccinated, we are able to do this again.